Okay. Okay, great. Hello, everybody. This is Evidence for You, broadcasting live via Google Hangout. Today we will have a discussion about evolution with the primary focus on DNA. But before I welcome our two main guests here, there will be a small chance for other Christians to join the Hangout later on. You can contact me on Google Plus or Skype if you want to participate. I have left the links in the description. So, without further ado, I welcome NephilimFree. Welcome. Okay. And then I also welcome Professor Dan Larhammar.
Hello.
Yeah. Hello. And for those who don't know you, Professor, can you please tell us a little bit about your work?
All right. So my name is Dan Larhammar. I'm a professor of molecular cell biology at Uppsala University in the township of Uppsala, just north of Stockholm in Sweden. My basic degree is in pharmacy, then I did my PhD in immunogenetics in the medical faculty, and after that I studied neurobiology, and I've been working along those lines ever since. And I specialize on the evolution of components in the nervous system. So my specialty is evolution of the vertebrates, and especially evolution of the genomes, the gene repertoires of vertebrate species.
Yeah, thank you. And then I will turn to NephilimFree, and you can talk about yourself. And after that, I will give you the floor, and you can start asking questions to Professor Dan Larhammar. And the less I will have to intervene, the better. Please be courteous.
Okay, my name is Evan Phillips. I go by NephilimFree on YouTube. I'm a Christian apologist. I'm a young Earth creationist. I became a young Earth creationist even though I was a believer in God by studying the evidences for creation. I do Christian apologetics, and I'm very pleased to be here and speak with Dr. Larhammar.
Just call me Dan, please.
Dan, thank you.
Yes. So, Evan, when you say young Earth creationist, how young do you think the Earth is?
I think between 6,500 and 10,000 years.
All right. And what makes you believe it's so young, despite the records of sediments and ice cores and tree trunks and other evidence?
Well, there are a host of evidences that provide a reason to believe the Earth is young. The decay of the Earth's magnetic field, carbon-14 found in dinosaur bones and coal and diamonds, the record of man having walked with dinosaurs, which is extensive, well recorded in human history, literally, in literature and legend, and in human art. Those are just a few.
But if one follows the dendrochronology samples from tree trunks, already those point to an age well over 10,000 years.
Well, I'll address that. First, let me mention sediments. You mentioned sediments. The sediments that cover the Earth are average 1,800 meters in depth across the continents. And the problem for the uniformitarianist is that the sediments these stratas are created by moving water that's known to hydrologic.
Yes, but please, can we take this step by step? If we start with the younger ages and the younger evidence, I think the first observation that is really striking that will take us beyond 10,000 years is the dendrochronology. So how do you explain the tree rings that take us well beyond that age?
I'll do that, but you mentioned strata first, so let me refer to that first. The strata of the Earth were created by moving water. That's been demonstrated to be a scientific truth according to hydrology experimentation. And this nature of the strata is that they have particle size distribution, clear distinct boundaries, and an absence of erosion between them going throughout the entire geologic column. Now, that means that there is no way for the uniformitarianist to squeeze their imaginary time in between one strata and another, because one strata below is created at the same time as the one above it. That means the imaginary time between them disappears. It does not exist. Now, as for tree ring dating has come under fire from a number of scientists. You can find university professors' web pages where they acknowledge the fact that tree ring dating is specious, because it's known that trees produce false rings, and many times in the lifetime of a tree, it's possible for a tree to produce many false rings. So I would say that there are no empirical evidences the Earth is of vast age. There's an assumption based on uniformitarianism, but no empirical evidence.
It is not an assumption, it's just counting the tree rings. If we go to the next level then, with the glacier cores, those give us layers that are hundreds of thousands of years. So that extends that record. And then we have the sediments, and then we have the geological layers with all the fossils that follow that chronology. So by putting all these together, there is little doubt that the Earth is much older. You mentioned carbon-14 and dinosaurs. Why on earth would one try to determine the age of dinosaur fossils with C14?
Well, first let me address what you just said. Back to the geologic column for a moment. The ice cores, for example. Is it your belief that an ice layer, that the various layers of ice found in a glacier, for example, are laid down annually? Isn't that the historical assumption of the uniformitarianist, and do you concur with that?
I'm sorry, I didn't quite hear what you said.
Okay. I was asking if you believe, you mentioned ice layers in glaciers, and
yes.
the historicity of that is that the uniformitarianists have historically believed that the layers of ice in glaciers were laid down annually, that each represents a separate year. Do you concur with that?
I'm not sure if there's one every year, but one can follow the strata in the ice cores, and by piecing them together, one get ages that are much older than the ones you assume for the Earth.
Okay, well, here's what I would posit as something to consider regarding ice layers. Ice layers are now known to be produced by storm events. A single snowstorm can produce a layer. And if it snows 1,500 times in Greenland during a single winter, you could have 1,500 layers. And that would be, according to the uniformitarianist of 1900, that would be 1,500 years. But it happened in a single year. There is an airplane in 1944. Some pilots crash landed a P-38 aircraft, fighter aircraft, on a glacier in Greenland because they ran out of fuel. 60 years later, they got wondering what happened to their aircraft, and they went back, and using sonographic equipment, they found that airplane 258 feet down inside the glacier under thousands and thousands and thousands of layers of ice. They actually retrieved that aircraft by melting the ice and pumping the water out, and it's now flyable. So 258 meters of layers of ice to the uniformitarianist would represent tens of thousands of years, but yet they were produced since 1944. So,
do you have a reference that describes the characterization of this finding?
Yes, the name of the aircraft is Glacier Girl.
All right.
The men who retrieved it and flew that plane have a website devoted to it.
All right. And there's been a scientific investigation carried out?
I don't know that there has. There have been many scientists who have written to the fact that layers of ice in glaciers do not represent annual depositions. However.
All right.
So maybe that's something you should look into.
I certainly will. Carbon-14 then. Why determine the age of dinosaur fossils with carbon-14? I'm sure you know the half-life of carbon-14, right?
Yes, approximately 5,670 years, roughly.
So, do you know how many years it can be used then?
Yes, well, if according to physical chemist Jonathan Sarfati, if there was a ball of carbon-14 the mass of the Earth, it would disappear in 1 million years. So I understand that the rate of its decay is quite high, in comparison to the imagined age of the Earth. But so that is a good question, though. Why date a dinosaur with carbon-14?
Wait a minute, I didn't quite get the mathematics here. How many years do you think this method is useful? Did you say 1 million years?
No. I'm saying I agree that it seems absurd to date a dinosaur bone with carbon-14, but the point of it is that since carbon-14 is found in them, it seems to argue that the dinosaur bone could not be so many millions of years old, lest there be no carbon-14 found in it. That's the point. I agree that no secular geologist would bother dating a dinosaur bone with carbon-14, but yet it has been done.
But the question is, how much carbon-14 is there in it? And where does that come from? Is it microorganisms contaminating the samples, or is it the age of the bone itself?
Well, contamination is often an argument used by uniformitarianists and evolutionists whenever, for example...
and creationists. We have the Shroud of Turin story, you know.
Okay. Okay, perhaps that one is true. But contamination is often used.
The younger ages have been contaminist. So it can go either way. By either side.
I would have to investigate that particular example, but I do know of examples where that it seems to be a habit of evolutionists to throw a flag up and declare contamination whenever something argues against their views. For example, when bacteria are found in a salt inclusion in strata believed to be 250 million years old and then revived, because of what we know about now about the half-life of DNA, they could not possibly revive. There would be no useful bits of DNA left in bacteria.
I'm sorry, half-life of DNA.
And yes, and so there could be no useful DNA that could cause a living organism to thrive after 250 million years. Therefore, so what the evolutionists did, of course, is throw the scientist under the bus. That is, well, he contaminated. And they do this often. Whenever evidence is put forth that seems to argue against their views on things, they often resort to this, well, it was contaminated. Well, it was contaminated. Well, it was contaminated. You hear that a lot.
But let's get asked why using carbon-14 for samples that one would assume could be much older. Do you know the span of the carbon-14 method? That is, for how long can one measure carbon-14? How old samples can be dated with this method?
Well, as I said, if a ball of carbon-14 the mass of the Earth existed, it would be gone in 1 million years. So I understand that there could be no carbon-14 in a dinosaur bone if it's 65 million years old.
In fact, the limit of the methods is much shorter time than that, because after some 5,000 years, there's only half left. After 10,000 years, there's 1/4 left. After 15,000, there's 1/8, and you halve the amount for every 5,000 years. And with the sensitivity of the methods today, it's very rare that one can use this method beyond 40,000 years, that is 8 half times. So anything older than that, this would be a useless method.
Well, then you would agree that if carbon-14 is found, because if it is dated with carbon-14 dating, and carbon-14 is found in the dinosaur bone, then it should not be there.
If it is a true component of the bone.
Right.
And that is the question.
Mm-hmm.
So using carbon-14 to determine the age of such old objects, or objects that one would have reason to assume could be that age. It's like sitting above the Challenger Deep in the Pacific Ocean and trying to determine the depth there with an instrument that can reach 1 meter down. Everywhere you put it down beside the boat, you conclude that the depth here is 1 meter, because that's how far the instrument takes you. Using carbon-14 for anything older than 40,000 years would be equally stupid, because the method doesn't work for older objects.
Well, I would agree that it would seem to be impractical if you believed that if outright that what you're attempting to date is as old as what you believe, if it is millions of years old. But the fact remains,
well, it's not a matter of belief. It's just it's a matter of checking other types of evidence.
Excuse me. That it indicates that there should be no carbon-14 in it. Whether or not it's some practical idea according to evolutionists or not is really, to me, irrelevant.
All right. Do you have some references that can inform me about the diners contain significant quantities of carbon-14?
I'll be happy to look for that.
Please do.
Mm-hmm. It might. I really don't want to dredge this down into it, because then you're asking me to dig around, to remain off the mic and dig around in my favorites for links to things instead of just.
No, I don't ask you to do it now. But if you could find those references for me, I'd be happy to take a look at them later.
Okay. All right. All right. So, if I could ask you a question now, I would ask you, why is it that you would believe the Earth is millions of years old, if there are so many evidences that it is not? For example, you mentioned the strata of the Earth. Now, it was not known in the days of Charles Lyell that strata were laid down by moving water. This idea of Steno explained it to us, and this idea was discarded basically by secular geologists for 100 years, 150 years, or actually more than that. But then when we come into the modern era, we find that hydrology experiments demonstrate that strata are created by moving water. So the Earth is covered, here's the evidence, the Earth is covered with deep strata laid down by moving water filled with rapidly buried creatures. We call them fossils. And this is a global feature. So I cannot imagine how somebody can know this and not understand that the Earth has been globally flooded, except to say that this part of the Earth was flooded at this period of time, and 50 million years later that part was, and 100 million years later that part was, and sort of break it up piecemeal, which is what the uniformitarianist geologists attempt to do. Isn't the evidence just this much clearer? The Earth is covered with strata, very deep strata created by moving water filled with rapidly buried creatures. That indicates to me a global flood, if anything does.
The evolutionary distances that I'm studying extend over 500 million years, the evolution of the vertebrates. And then we have a different type of evidence that also supports a time scale extending over these vast ages, and that is the movements of the continents with a continental drift. And by measuring the movements today, we can extrapolate backwards in time and see how the continents used to belong together into, for instance, one supercontinent long ago. And the interesting thing is that both the distribution of fossils and the distribution of species agrees very well with the movements of the continents, and especially when it comes to formation and disappearance of land bridges. There are a couple of animal groups that have been studied very carefully, and one can see that the spread of species agrees very well with land bridges between the continents. Now, the type of evidence that I'm looking at, as I said when I introduced myself, is the genomes of organisms. I look at the genes and how much they differ from one another in different animal groups. And it turns out that this is a written record of the history of the species. So even if Charles Darwin had never lived, we would be equally convinced that there has been an evolutionary process just by reading the sequences of the genes, because it agrees so well with the evolutionary relationships that he and many other scientists following him have deduced from comparisons of fossils, comparative physiology, comparative embryology, and several other fields in biology. So by comparing the genes totally independently, we can calculate an evolutionary tree that shows the same relationships as the fossil record. So evolution has been discovered not only twice, but actually multiple times from different branches of biology, biogeography, etc. So evolution has overwhelming evidence from many different branches of science.
Well, I would argue that your idea that phylogenetics matches the fossil record is completely erroneous. The fossil record, the fossil record shows no evidence of transition. Numerous scientists, highly accredited, even the world's most famous paleontologists and geologists, have made admissions that the expected forms are not there. But you mentioned, but I would say this. What I notice when I have discussions with evolutionists about these things is I notice that, for example, a moment ago we were talking about the strata of the Earth, the nature of the strata of the Earth. If we were to just negate all other arguments and stick just to that, and I don't expect us to do that, I don't want to do that, but if we were to just do that, this discussion would come to an end, because the evidence is 100% on my side. Because strata cannot be produced by long deposition. They are produced by cataclysmic moving water is what explains the geologic column. But I notice that then what happens with evolutionists is then they use one assumption to back another assumption to back another. So then you go to, well, the continental drift, and then to phylogenetics, and then to the geologic record, the rock record of life, and one thing always supports another within the mind of the uniformitarianist. See, and that is what I think evolution theory is a large package of assumptions based on the paradigm that life was not created and that nature, the Earth itself, is the producer.
As I was just trying to
everything must fall in under that paradigm. It must. And so all things support that in the mind of the uniformitarianist. But if we were to dissect any given particular subject, whether it's genetics, phylogenetics, the fossil record, the strata of the Earth, the geology of this planet, and strictly on that, and compare evidences after another, we could do it hours after hour, and the evidence would be 100% in my court.
All right, let's do that with the genetics then.
Can I just interrupt for 1 second here? I please will want to remind you before it escalates that please be courteous and please wait till the other one has finished. And if it happens to be that you cannot agree with the topic, we could at least agree to disagree, and then we can move on. Okay?
Of course.
Okay.
I would like to address the idea that phylogenetics matches the fossil record, as you said, though. Can you tell me, please, what is it about the fossil record of life on this Earth that gives us reason that supports the phylogenetic idea and the comparative analogy, homologous structures idea that you touched upon?
As I said, the history of the genes correlates very well with the fossil record. If one follows the fossils as they have been reported in different layers. I had the pleasure just 2 weeks ago to attend a conference organized by the Royal Swedish Academy of Sciences and my colleague Professor Per Ahlström, who had invited some of the world's most prominent paleontologists working with the origin of the vertebrates and some of the most important and successful comparative genomicists comparing the genomes of presently living species. And it turns out that together these two data sets can disentangle the early stages of vertebrate evolution to an extent that was probably impossible to imagine just a few years ago. The details they can see in the fossils with the modern physical methods is quite extraordinary. They can see not only the bones, but also the traces of the soft parts, like blood vessels and parts of the nervous system. So we can see which structures are there, and some of those structures and their origin agree with the appearance of genes that are involved in these processes. So if we look at the genes, we can deduce the different radiation steps or divergence steps for the different lineages in vertebrate evolution. You said that genetics was on your side. In what way? Can you please explain?
I'll be happy to. But first I want to address it. You said early vertebrate evolution is evidenced by the strata. I would remind you of the Cambrian explosion, which is a conundrum for the uniformitarianist, that so many body plans and phylum appear in a geologic spit in a bucket. That seems to, it's actually turned the Darwinian tree life upside down, especially with the Cambrian fossils being discovered in China today.
No, you're not giving a fair description of the situation.
You mentioned also soft tissue preservation in some of these fossils. I would point again to the idea of fossilization, that should we have, like the baleen in the whales in Argentina, it's not the only way that these soft tissues in creatures could be preserved is if they're buried rapidly. Now, I don't know where you're going to squeeze your imagined geologic time in where an organism has been buried so rapidly and sealed away from oxidization and predators, scavengers, that its soft tissues would be preserved like jellyfish in the Cambrian. So I would say that that record of creatures argues against uniformitarianism, not for it. But to address what you said about genetics, I would say the fact that we even have a DNA molecule is empirical evidence that life was created. Every living thing on this planet is defined by information which is organized to conform to linguistics laws, possesses in chemical function the human language properties of phonetics, semantics, punctuation, syntax, and grammar, and possesses algorithmic input and output function. I would say that information, algorithms, and linguistics are products of an immaterial mind, and therefore it's empirical that all life was based on was produced by a mind, designed by a mind, because I think it's illogical to believe that information, linguistics, and algorithms can be produced by chemical process.
I don't understand what you're talking about.
Well, you do understand that genetic information, that there is algorithmic input-output processing, copy-paste insertion, these kinds of things in the genomes of the
You make it sound like a computer, but we're talking here about a molecule that is being replicated with lots of errors happening all along. And you must be aware also that a large proportion of the genomes of many organisms do not carry information. They are just spacers in between those regions that have biological functions.
But the codes for proteins by themselves are prescribed information. Prescriptive, not descriptive. It prescribes exactly the chain of amino acids in exactly what order to produce a functional three-dimensional molecular machine. That's information in my book. And I don't see how nature is capable of producing algorithms or chemical processes that can be likened directly to phonetics, semantics, punctuation, syntax, and grammar, like a pause or stop codon, these kinds of things, go-to statements. How is it possible for mere chemistry to produce these things which we know are products of intelligence?
Because these are self-replicating molecules. The part of the human genome that encodes proteins is approximately 1%. There's a little bit more that is under selective pressure, probably of the order 5 to 8, possibly 10%, but the rest is not. So the rest is what is usually called junk DNA. Now, I'm perfectly aware that this term has been misinterpreted, sometimes deliberately, by many, including some scientists, to be honest. But what we mean by that is that there is no selective pressure preserving these parts of the genome, and they have no specific functions that can be ascribed to them. Now, a large proportion of that is nevertheless well conserved between closely related species, and there is no functional reason why that would be preserved unless these species that are more closely related share a more recent common ancestor. And we have overwhelming evidence for this from a huge number of species. And one of the most familiar examples is for sure the relationship between humans and chimpanzees, where 99% of the entire genome is identical between these species. Or down to 95%, depending on how you count the pieces that are missing in one species or the other. So, yes, I'm sorry.
I'm sorry. Could you, I don't think that you have your chat open, Dan Larhammar.
My what?
Your chat. If you hover your mouse to the left, you see this chat option.
All right, where's that? In the left-hand margin?
To the left, on my window, it's at the top.
There's the second symbol says chat.
Yeah, if you click on that, you will see that I post some things on to the right here. That could perhaps help you to either move on or so to speak.
All right. Yes, I can see it now.
Yeah, great. Just so you're up to date. I think it's Evan's turn. Am I right?
I think so.
Yeah, great.
You mentioned junk DNA. I would say I would ask if you're not aware that the idea of junk DNA has been pretty much thrown out. That's an old idea.
It has not.
Well, I would differ with you on that. There are papers in the journals, even Science today, various secular science sources that argue against the junk DNA paradigm. For example, Richard Sternberg, a geneticist, states, and I quote him, "We now know that based on genetic studies, based on experimental studies, that so-called pseudogenes don't code for proteins, but they have important function in chromosome biology, such as the way chromosome fiber folds, and they may also code for RNAs, which may be spliced and regulate other RNAs that make other proteins." Dr. John Greeley of Albert Einstein College of Medicine in New York states, "It would now take a very brave person to call non-coding DNA junk." And the list of these references goes on and on. There are a great many scientists today who acknowledge that studies have found that areas of the genome that have been claimed by evolutionists to be junk are known not only to code for proteins, but to have critical function in embryonic development in the human being. For example, areas that were claimed to be junk are now known to produce the human placenta. And so this is very critical to the development of a human being. Richard Sternberg points out that what are called endogenous retroviruses found in quote-unquote junk DNA are so critical to human development that at stage 4, when the human gamete splits, when you get to the stage of only 4 cells, that if this endogenous retrovirus is not present, there will be no human being. And this is found in what was called junk DNA for the last several decades. So I would say this junk DNA, I could provide numerous references, almost all of them from secular sources, that this junk DNA paradigm has been overturned by modern, more recent genetic studies, and in fact has held back genetic science for 40 years. Because of the evolutionists' belief that the majority of our genome was junk, they refused to investigate it and concentrated their studies almost exclusively on the protein coding area, which is, as you say, 1%, some estimates 3, 80% of the genome and held back genetic studies. So the evolution paradigm, because of the belief that the majority of our genome was actually junk, which is now overturned, has actually held back science.
Not at all. This is a distortion of history. Scientists have been studying RNA coding genes that do not give rise to proteins for decades throughout this time, and many of the regulatory RNAs that have been found are a tiny fraction of the small fraction that constitutes protein coding genes. So we are talking about a fraction of a percent when it comes to these small regulatory RNAs. It's interesting that you bring up the retroviruses because they are such a beautiful example of evolutionary relationships, because one can trace the spreading of these in the evolution of the primates, for instance. In fact, just by looking at repeated parts of the primate genomes, one can reconstruct the whole phylogenetic tree of the primates. So that's been a very useful type of genes to sort out evolutionary relationships. Now, I am aware that some scientists in the ENCODE project have claimed that a large proportion of the human genome has a biological function. But if you go back and read carefully how they define a function, you will find a very loose definition. In fact, it's almost circular reasoning. What they call function is something that happens to be transcribed or turned into RNA. Now, more careful studies have shown that it is a very low abundance of such RNA transcripts from the human genome, and the further away you get from the protein coding genes, the less there is of these long non-coding transcripts, which suggests that they are just a byproduct of the transcription processes, because regulation of transcription is by no means perfect. It would cost far too much energetically to have the rest of the genome completely switched off. In fact, we have leakage of transcription in many different tissues, although they don't use those particular genes. So there's been a quite intense debate after the rather scandalous publication from the ENCODE project. I believe they have to justify the enormous funding they have received by claiming to have found additional functions, but they have to be taken to task to show that all these things really do have functions. Many scientists are very, very skeptical. Finally, one must also distinguish junk from garbage. There is a distinction. Junk DNA can keep evolving by chance, and once in a while a new gene may arise, and we have a few examples of that are being characterized. New genes arise from the junk. The garbage, on the other hand, are useless genes that have been so destroyed that they cannot give rise to any functional product. And I could give you several examples. I'd like to come back to that in a moment, because this is one of the strongest pieces of evidence for evolutionary relationships.
I don't know where you're getting this idea, because 84% roughly of the human genome is now known to be transcribed. That goes back at least as early as the mid-1990s. So if it's transcribed by the cell, this indicates that it is function, that it's purposeful.
No, I was just trying to explain that it's not.
Well, if it's not, why is it transcribed? And why
I just told you, because it would cost so much for a living cell to have the rest of the DNA switched off, and all the processes would become very slow if there were blocks sitting there trying to keep the polymerases off from those pieces.
I think it makes much more sense to say that if it were junk, natural selection should have removed it. Especially if natural selection is the genie that evolutionists believe that it is, it makes no sense to believe that if evolution is producing various creatures, new structural designs and living things over these vast ages of time, and is capable of doing such marvelous things by selecting for genes based on their usefulness, I think it demonstrates quite clearly, I think, that when scientific evidence comes up that argues against the evolution idea, the evolutionists are quick to get on board with something that they see that seems to imply their theory is correct, but then completely disregard something that doesn't. For example, if you believe that mutations occur which duplicate genes, and then these genes are acted upon by subsequent mutations to now form new information which codes for new functions and new features and new anatomical designs in living things, and natural selection somehow, you believe, selects those, but then and deselects the negative mutations, but then for some reason it can't deselect 98%, 97% of junk in the DNA. I think that's really, really stretching the imagination. That's just picking and choosing to the highest degree. That's like saying I'm willing to believe that natural selection can select for these little changes over all this time, but can't select out. It somehow deselects negative mutations, but gee, apparently it didn't, because it left 98% of the DNA as junk. I think that idea of junk DNA is utterly preposterous. If it's transcribed, it must have function. If it's preserved, you would, even as an evolutionist, if they were, I think, being true to their own theory, would have to argue that if it's preserved, it's because natural selection selected to keep it, because it's functional in some way.
Can I give you a concrete example? The human gene for one of the dopamine receptors is transcribed, but since that gene, like so many others, have multiple non-coding regions within it that have to be removed before the protein can be made, this requires a special mechanism called splicing. Now, it turns out that more than half of the RNAs that encode this dopamine receptor are misspliced and give rise to a totally useless product. And in fact, there are many genes in the human genome that are misspliced, or there are genes that give rise to RNAs that cannot be used at all. And I'm sure you're aware of some of these. The classical examples that I believe many creationists know about too is the gene for production of vitamin C. As you know, humans cannot produce this crucial vitamin, but we have the gene for it, or at least we used to do. We still have a piece of the gene that shows that our ancestor once had the ability to manufacture vitamin C. So apparently we have had this ability. Now, the really interesting thing is that this non-functional gene is also found in the genome of a chimpanzee and in the genome of the gorilla. And the gene has the same deleterious mutations in these species, showing that the gene was destroyed in the common ancestor before it diverged into these three species. And this is just one example. I could talk for hours about examples like these, not only among the primates, but also in many other evolutionary lineages where non-functional genes are inherited in different species because they come from the common ancestor.
So I would say you look at genes between two different species and you assume ancestry because you find the same sequence of amino acids. I mean, not amino acids, but nucleobase pairs or codons, and assume then heredity, the relationship between the two. It argues for me a common designer. And I think it's perfectly plausible that a human being could have the same gene as a chimpanzee for the same purpose, because they were designed by the same creator, and both could be damaged due to genetic entropy. I don't see any problem with that in creation whatsoever. However, I would point out that if again, I would just say that if natural selection is the preserver of what's good and remover of what's bad in DNA, and it has millions of years to do this, it should not preserve the majority of the DNA if it were junk. And since we know it's transcribed, and we know it has function, even critical to human embryonic development, that certainly can't be considered junk, can it?
There are millions of ways whereby a gene can be destroyed or disrupted. Now, the gene encoding the enzyme that should manufacture vitamin C has been destroyed in the same way in the species I mentioned. So if there is a God that designed this, it must be a very careless and sloppy God that not only makes mistakes, but makes the same mistake over and over again in these different species. No, that's a fairy tale, of course. A much more reasonable explanation is that the mutation happened in the common ancestor of these species, and then the species diverged from one another.
Well, because of the nature of the sequence itself, it's very plausible that the mutation, which caused a particular damage to one gene, would happen in the same way in another. That's not implausible.
There are many other species that also have lost this gene encoding the enzyme that makes vitamin C. Bats, for instance, and guinea pigs, but they are more distantly related. And lo and behold, their mutations are different. So their genes have been disrupted independently. But if there had been a designer, wouldn't he have designed their mistakes in the same way? Again, the evolutionary relationships tell a much more coherent story where the phylogeny of the species correlates very well with the time points of these mutations. And this is just one of the many pseudogenes, non-functional genes that we have in our genome. We have approximately 20, perhaps 22,000 functional protein coding gene, and depending a little bit on how you count, we have somewhere around 12,000-15,000 non-functional genes. So significant proportions of the genes do not work. And then I still haven't counted the 1 million copies of another RNA coding gene that went berserk in the primate genomes and actually constitutes 10% of the human genome, a non-functional piece of DNA encoding an RNA molecule that is no longer transcribed. And in many other evolutionary lineages, we see similar developments where genes have spread and now comprise a large proportion of the genome. And this actually explains why there is so much junk DNA in many different species.
Yeah, I completely disagree. I see that the junk DNA paradigm has been thrown out by the discovery of critical function in that portion of the DNA. And to say that just because we don't know the function of much of it, does not mean that it's junk, because I see as an ad hoc explanation based on the evolution paradigm. I find no problem whatsoever in finding sequences of information in the genome of one type of life and in another that could be considered evidence of a common ancestor. You mentioned bat evolution. I would remind you, bats rather, I would remind you that there's no support for bat evolution in the fossil record whatsoever. They inexplicably appear without any potential ancestors. So that, like every other creature in this world, is not supported by the fossil record of life. There are no ancestrals that we can point to. We cannot put together any lineage of fossils, any 5 or 10, or even 3, and show anatomical change take place. I say it happens only in the imagination of the evolutionist. You don't find it in the fossils, and therefore the assumption of phylogenetics is based on assumptions that because we can find the same sequence of information in a genome of one kind of life and another, that it must be evidence of common ancestry. But I would remind you of the tremendous difference between different species that are claimed to be have common ancestry, and so there are tremendous genetic differences as well. For example, the endogenous retrovirus claimed to be ancestral between humans and chimpanzees, which is responsible for human development at stage 4 in the embryonic development. It's not logical, is it, to believe that the genome of a virus could have the information necessary to regulate the development of a human being? Well, I don't see that as logical. That's, to me, like believing that a 2-page pamphlet on how to assemble a tractor could have relative information necessary for the design of the tractor and all of its components. The regulation of the workers and the order in which they assemble the tractor, I don't find that a plausible idea at all. And I would ask you, can you tell me why you would believe, though, that these things, this human language and linguistics and information that we find in the genomes that defines every living thing on this planet, what is a plausible explanation from naturalistic process for the production of these things, which are products only of intelligence?
We do not find a language in 99% of the human genome. It's only in the protein coding genes, and that has been selected through a gradual process with the synthesis of amino acids into proteins. And we are using pretty much the same code as bacteria. There are only very minor differences in some special lineages of organisms. Otherwise, we all speak the same language when it comes to protein translation. But when it comes to the genome, you must separate function from transcription. It's known since decades that there is leakage transcriptions. When a gene is transcribed, it starts somewhere around the beginning, just before the coding region, but transcription continues way beyond the end of the protein region. Of that transcript is not used. It's just chopped off and degraded, and that's a large proportion of the genome.
If it's not language, why is it that scientists have recently discovered a second code in the DNA that rides atop the other codes? For example, the codes which the sequences which code for proteins is now known to be governed by sequences of information that overlie them, sharing base pairs with the underlying codes that code for proteins. This is regulatory information built on top of the other codes. It's like if you had a book that says, "In the beginning, God created the heavens and the earth, and the earth was void and without form, and darkness was upon the face of the deep." And in order to be able to read that sentence, you have another sentence that says, "In the beginning, darkness was upon the face." You see what I'm saying? Sharing letters, sharing words within that same two sentences is yet another code that tells you how to read the one below. Now, my example was a poor one, but I think you understand what I mean. So I would ask you again, if this information can be produced by natural processes, by mere associations of molecules, why is it logical to believe that any of that information could arise by natural processes? And at the same time, if your idea of block mutation, duplication by block mutation, whatnot, is what the builder of genomes, if that be true, could you explain why it's logical to believe that as DNA is being increased, the genomes of living things are being built by duplications of existing genes, some individual ones, and then entire gene sets? At the same time, this same process, which happenstance by random mutation, is building genomes at the same time building a code on top of them using those very pieces that are duplicated to produce a code that tells you how to use the one below? Could you explain that idea to me?
I'm not sure what you mean. A second code on top of the protein coding code.
Oh, okay. Well, it's recent discoveries. I'll give you a link here. There are many you can find. This one here. This is from ABC Science. Excuse me. And they state Dr. John, I hope I pronounce his name, Stamatonopoulos, University of Washington, Associate Professor of Genome and Sources in Medicine, states, "We know that this basic assumption about the reading of the human genome missed half the picture. Many DNA changes that appear after protein sequences may actually cause disease." Well, oh, wrong statement. In other words, they're arguing now, they've discovered that there is a second code that rides atop the ones that produce proteins that is regulatory, that explains how to regulate, how to use the ones below. Here's a link from Science Daily. Let's see, I think, yes. This is from Science Daily. They state, University of Washington, scientists have discovered a second code hiding within DNA. The second code contains information that changes how scientists read the instructions contained in DNA and interpret mutations to make sense of health and disease. Genomes are genetic code.
I'm sorry, I lost you there for a couple of seconds.
Okay. Well, basically,
hello? I can't hear you.
You can't hear me.
Hello.
Hello. Do you hear me now, Dan?
Yes, I do.
Okay. So this source says, and I'll just read one sentence from it. They state, scientists have discovered a second code hiding within DNA. This second code contains information that changes how they read DNA. What it is they've discovered this. The fact that the genetic code can simultaneously write two kinds of information means that many DNA changes that appear to alter protein sequences may actually cause disease by disrupting gene control programs, or even both mechanisms simultaneously. In other words, a mutation which damages a code for a protein is also at the same time damaging a code that tells the cell how to use that code for the protein, because that code is written on top of the other code. And this is information packaging of the highest technology. This is information written upon top of information sharing the same base pairs with the information below it. And so this is a profound argument against the idea that random mutation is a builder of good things in living things, as the evolutionists claim. For if mutation is damaging one sequence of information, it's now known to likely be causing damage to yet another, not only because genomes are now known to be overlapping and nested information sequences and base pairs shared between more than one sequence of information at the same time, but there is now known to be a code writing the top of other codes. And so if mutation damages one below, it's damaging the code that tells the cell how to use the one below at the same time. And this is a profound implication for evolution theory. I would say it refutes it by itself, because it's not plausible that random genetic mutation could build both codes at the same time by developing genomes by these processes of random mutation. It'd be like writing a book and with mutation, and then at the same time creating a code system written on top of that book on how to read it. I don't see that as logical or rational, that evolution could do such a thing by chance associations of molecules. Maybe you could address that. Tell us why it's logical to believe the mutation can build genomic information, at the same time build genetic code that spans beyond that sequence and is shared by base pairs over other sequences of information at the same time, and build both and both be functional at the same time.
It's been very difficult to follow your discussion because I lost you several times. But in the meantime, I looked up this reference by Stamatoyanopoulos, and I assume that you are aware that he is very controversial and has been strongly criticized by other scientists for the ENCODE project in particular. This second code you're talking about, I have to be honest and say I wasn't even aware of this. This is nothing that has been discussed in the genome circuits where I'm active. So clearly it has not made much impression in those who work with the genome. But I'd be happy to take a look at that and see what he actually means, because from what you read, I do not understand what he's trying to say beyond regulation of gene expression.
Well, what sounds to me like you're doing is, and I mean no disrespect, but it does sound to me like what you're doing is what evolutionists often do. Whenever profound evidence against their views is discovered, they invariably throw the scientist under the bus, call him a rogue and whatnot. And then we find 10 years, 20 years, 30 years later that their idea is not rogue, such as this junk DNA idea, which even Francis Crick himself acknowledged is false. So this was the case with the Italian scientist who found bacteria in the salt inclusion, claimed to be 250 million years old. They threw him under the bus because they claimed it was contamination. You know, anything that argues against evolution, you know, throw it under the bus. This is how evolution theory holds back science.
Now, hold a minute. I know the junk and garbage story very well, and these terms were introduced by Sidney Brenner, the Nobel laureate. And this discussion is still going on, and it's the ENCODE project with Stoyanopoulos that have been strongly criticized by many members of the scientific community.
Well, for what reason?
Because the reason for that is that they mix up transcription and function. And I just explained to you that many genes are transcribed rather inaccurately, and especially far beyond the useful part, there is still something that is transcribed which is totally unused. Now, by the ENCODE project, this would be called a function, but for any normal biologist who really looks for the functions, this would just be a leftover, a remnant, which is useless. Yeah. So, and there have been some really excellent papers written on the distinction between transcription and function, and I'd be happy to send these links to you, because I think they will help to make you understand the distinction between this very loose and sloppy definition that the ENCODE project is using and the stricter biological definition that everyone else is using. Now, if we come back to the
sloppy.
Yes.
Any scientist that produces information, new discovery that doesn't comport with evolution theory is doing sloppy work, right?
No, that's not what I'm saying.
Please. I mean, okay. I heard that Don Larhammar used sloppy, but I've heard you use the term imaginary time for the millions of years. So please be courteous and move on. And if you once again, if you cannot agree on a topic, we could please move on. Okay, thank you.
Let me change the word sloppy to loose definition, but that means the same thing here. The ENCODE project have a very loose definition of function. They mean anything that is transcribed, and that's not a way to increase our knowledge. It's just a game of words that is totally useless.
So, but isn't it true that transcription is a critical function? And so why would you dismiss the fact that so much of the genome is now known to be transcribed and still consider it junk?
For the third time then, gene regulation is not very accurate. Parts of genes are transcribed way beyond the piece that is used as a protein. There's a long tail of the transcript that is not used. In fact, almost every transcript in the genome has to be trimmed at the end because the polymerase ran too far. And as I already described, the further away from the known genes we go, the less we have of these long transcripts, indicating that they are probably leakage transcription. Not all of them. I'm not saying that all of them are junk, but many of them are probably just leakage transcripts. And I gave you an example of useless DNA with a dopamine receptor. I could speak to you for hours with examples of genes that are not transcribed or spliced properly and give rise to useless products. Many of them do no harm, but sometimes a gene due to a mutation will cause harm because it messes up the machinery of the cell in this way, and there are diseases known which are caused by genes that have such mutations.
How much
so
of
a
the
large
genome,
proportion of the human genome. Yes.
How much of the genome which you believe is junk do you believe has no is not known that what is transcribed? How much of it do you believe is shown to have no function? Of the 84% of which is now known to be transcribed, which you believe is junk, do you know, have an idea, an estimate based on anything published?
There are many estimates in the literature. 1% encodes genes. Another 5 to 8%, perhaps up to 10%, is under selection. Much of that probably because it's involved in the regulation of the coding parts or for other reasons. So the remaining 90% or slightly less is under no selection. However, that doesn't mean that all of that is useless, because we also have something called the distance effect. So some DNA works as spacers, but the structure of that DNA probably doesn't mean much at all. It could be any piece of DNA sitting between two used genes, but the spacer is necessary to keep them apart so that the regulation doesn't interfere between genes. We have some genes whose regulation is overlapping, that is, one gene.
Well, then that spacer has function, does it not?
Yes, but it's not the exact sequence of the DNA. You can throw in any piece of DNA virtually in that region and it will serve the same role. So that doesn't mean that there is a strict definition for that region.
I guess what I'm asking you is how you know that, since now we know that 84% roughly of the genome is transcribed. What I'm asking, I guess, is how much of that 84% which is transcribed, when it is transcribed, produces useless products? Do you have an idea of that?
Well, inferring from these other estimates, that would probably be, if we take away, my guess would be perhaps 80%.
80% produces, and when you say useless product, exactly what do you mean by a useless product? An RNA that has no function?
But it doesn't.
Is that what you mean?
Yeah. But then you must be aware that among those 16% that are not transcribed, some parts actually do have functions. So this is now why this transcription.
I'm sorry, go ahead.
Can I continue?
Yes, I'm sorry. Go ahead.
This means that this transcription criterion is not a very useful way to describe function, because some of the regulatory regions of genes are not transcribed themselves. They sit in front of the genes and determine when the gene shall be switched on or off. Other parts of the chromosomes are necessary for chromosome segregation and chromosome packaging. They need not be transcribed, but they are still useful. So transcription and function are not equal entities. In fact, there is a huge discrepancy between those two terms. And this is what several papers have now sorted out for the ENCODE people who got totally mixed up.
So of this, you say a large percentage of what is this 84%, which is transcribed, produces a useless product. How much of that, how much of this product, of this 84% of the DNA, has been studied in studies and shown to be of no use? Instead of how much of this is assumption and how much is verified by study?
We can equally turn the question around. Those who claim that this is functional, how do they know? How well have they studied these 84% to claim that it can be functional?
So is it safe to say then that your belief that it is is of no function is based on is not demonstrated by study, but is based on an assumption then?
No, it's not an assumption. It's based on evidence. If we look at the products that and we compare the selection. So there's a lot of circumstantial evidence as well as direct evidence.
Okay, I didn't hear
Have I
most
lost
of what you
connection?
said. Yeah, you were coming and going. I really didn't hear much of what you said. I'm sorry.
Yeah,
I had the
I
same
things
problem
got,
before.
yeah,
did you? Yeah.
that seems to be sort of a breakup when Professor Don was speaking. Uh, Perhaps if you can turn down your bandwidth if you hover at the top of your window. You can
Yeah,
turn
I think
down,
I'll turn it down one
Oh,
click and see if that helps.
yeah, or perhaps you can turn off your cam altogether. But
Yeah, that makes the biggest change, is
yeah,
turning off the
it
cam.
does.
But I hear him fine now. Maybe it was just temporary.
Yeah, maybe it's temporary. I don't know how this works, but I have turned off every window on my
Okay.
computer, so it couldn't be from here. So
Dan, I'm sorry that I didn't hear what you said. Excuse me. I asked you what percentage of this, how much of this presumed to be junk DNA, which is transcribed, which is now known to be a very large percentage of the genome, is based upon an assumption, or how much of it is actually shown to be producing a useless product, is actually verified by study. And I did not hear your response. I wonder if you'd be so kind as to respond again to that,
yeah.
because
I began by saying that that question can be turned the other way around, and one can ask those who claim that transcription means function, how much have they studied all these 84% of the genome that is they claim is transcribed? How can they know that all this has a biological function? The burden of proof is on the claimant. Now, there is a lot of circumstantial evidence that much of that does not have a function, and this is based on what we know is under selective pressure, not only between species, but also within populations. We can see where the variation is more abundant and where the variation is more scarce. Where the variation is scarce, that implies a function, and in some cases that has been tested also.
Okay, you said the burden of proof, that is, I take it from your answer that very little of this, what is called DNA, junk DNA, which is transcribed, has been shown by study to produce worthless products. I think that can be easily gleaned from your answer. And then you say, so what I see is, because we don't know the function of it, we just declare it junk. Then nothing has changed. That's what evolutionists have been doing to the majority of the genome all this time. And we continue to find more and more function in this area, large percentage of the DNA that's considered junk. You said, as regarding this, that the burden of proof is on the claimant. Well, I would say the evidence is clear. 84% of it is transcribed. It's now known that some of it is critical to human development embryonically, and some of it is known to code for proteins that was not known before. The simple fact that we have not studied the majority of the DNA with one study after another to know what this sequence of information does and what this cluster of information does, and is not reason to believe that it's junk, considering what we have discovered about what little has been studied. And I just don't see that as good science, to outright claim that most of the DNA is junk, even though it's transcribed. And we continuously find more and more critical function in it. Just to declare it junk because you believe evolution theory, I just can't fathom that, Dan. I don't know why you would do that. Why would you take a package of information that's three-dimensional, overlapping and nested, algorithmic input processes, linguistics properties, code for which has regulatory codes on top of regulatory codes, and then just declare the majority of it is useless because we simply have not studied most of it and don't know what it does, even though we know 84% of it is transcribed. That to me seems like a terrible way to approach science. I cannot fathom approaching science in such a manner as that.
Hello, are you still there?
Yes.
Hello.
yes, you're heard. It's your turn.
Okay. Yeah. your voice kept disappearing, so I'm not sure I understand what you were trying to say, but it sounded again that you were equaling transcription with function. And I'm not sure how many times I need to explain that this is something different. And there's an extensive literature on this now, so please study that literature and try to understand the distinction between transcription and function.
Yeah,
Now,
I will.
with the linguistic properties, I think it's been the effort of scientists to explain the genetic code as a language that have sort of seduced many scientists and lay people as well, but even some computer scientists, to look for intricate languages in DNA. And I have actually been involved myself in debunking some of those claims, because they were found to be present also in completely randomly collected artificial DNA sequences. So much for those languages. Now, this new language you talked about that was presented 18 months ago, maybe something else. I don't know about that, and I'll be happy to look further into that. I cannot comment on that right now, but it seems to me like this is the regulation of genes, in which case it is nothing new, but I may be wrong. Now, the junk DNA in the human genome, somewhere around 90%, means that our genome is fairly large, about 3 billion base pairs in each cell. Do you think that human beings are complicated species, or are we among the simpler ones?
I say human beings are exceedingly complicated. I don't know of any organism, even a single cellular organism, which is not exceedingly complicated. I would ask if I would point to what are called pseudogenes. Pseudogenes have been also considered junk DNA, but now we're discovering that these have function as well. Excuse me. And so that's just continuous discovery of function in this material. So I don't see calling it junk good science.
Can I give you the numbers, please? We have, I already explained, we have 1 million pseudogenes called the allorepeats from the 7SL RNA in primate genomes. 1 million pseudogenes, 10% of the genome. We have 20,000 functioning protein coding genes and somewhere between 10 and 15,000 pseudogenes for those. Now, a couple of those pseudogenes have been found to be able to interfere in some way with cellular processes. But this doesn't mean that all of them do. That's a bold extrapolation. One has to look at the numbers and not be carried away with wild extrapolations. What I was trying to get at when I asked you if you think a human cell is complicated when we have 90% junk DNA, is that we have the 3 billion base pairs in each cell. There is a species of newt which has 120 billion base pairs in every cell, 40 times more than a human being. Even a tree like the Norway spruce has 20 billion base pairs in each cell. Mm-hmm. Are they much more complicated than a human being if they need so much what we call junk DNA, but you may be calling functional?
Because we don't know what the function of it is, it's not reason to call it junk. That wouldn't be a scientific approach to it. Is study to find out what it does. That would be scientific. To declare it junk because you haven't studied it or know what it does, that's not scientific approach. I wanted to ask you about, you have said that about the 2R hypothesis, I quote you, a further entire genome duplication is believed to have occurred in the ancestral fish lineages approximately 320 to 350 million years ago. as well as more recent independent tetraploidization events, most not exclusive, in particular teleost and amphibian lineages. You said the majority of duplicated genes become pseudogenes and are deleted, but some may evolve to encode components with new functional roles. So apparently, from what you've published in the journal, you are a proponent of the 2R hypothesis, and that which has been highly, you mentioned things which are highly questionable in science. I would say that the 2R hypothesis has been a very sketchy idea from the start. It was argued against as far back as 1982. And I would consider this. As for pseudogenes, Dr. Richard Sternberg has stated, we now know based on genetic studies, based on experimental studies, this is real science he's talking about, no assumptions, that many so-called pseudogenes don't code for proteins, but they have important function in chromosome biology, such as the way chromosome fiber folds, and may also encode for RNAs which are spliced and regulated and regulate other RNAs which make other proteins. So there is increasing discovery of function in these pseudogenes, which are claimed to be useless leftovers of evolution.
And how many examples does he provide?
He does not provide a number, but he says, he does state, we now know based on both genetic studies, so he's referring to two different studies which provide evidence that what are considered pseudogenes are increasingly discovered as having important functions. But I wanted to ask you about this 2R
No, wait a minute. Now,
hypothesis.
again, the numbers are very important.
Okay.
We're talking about 15,000 pseudogenes, and he gave two examples?
He cites two studies.
Yes.
Yes.
And
Okay.
and which genes are they?
He did not state.
All right.
Mm-hmm. But what he's pointing out is
actually this is strong evidence with such meager references.
Well, these and the other things that I point out is that such as examples I've given about embryonic development and whatnot is evidence that the more we do study this area considered junk, the more we discover function, and that's so. Excuse me. A scientific approach to it would be to say. It's more logical, I say, to say we don't know what its function is than it is to declare it junk because you haven't studied most of it. That is not to me a scientific approach.
It's not a scientific approach to extrapolate from 2 to 15,000.
There are more than two. There are many examples of function discovered in what has been called junk DNA. The list continues to grow. I'm sure you're aware of that.
Others have been retracted of those initial examples. They were found to be artifacts from experiments.
All right.
So it goes both ways.
Yeah, I'm going to have to side with the continuous discovery of function and the idea that it's not scientific to declare it junk because we don't know what it does. When we do continue to find new examples of function. But I wanted to ask you about
but then if you postulate, you know probably the onion hypothesis, which was sort of what I was trying to explain a moment ago, when there are some species that have so much more junk DNA than a human being, both a Norway spruce and a newt, and many other species have much more junk DNA than humans. Why would they need so much of it?
Well, that's what we need to discover. That's what science is about. That's what we should. That future study may discover the function of it. But just to declare it junk is not scientific.
It is bold to argue that it's not junk when there is no evidence. But as I've tried to explain, there's a lot of evidence suggesting that it is junk because much of this is repeated segments like retroviruses, transposons, and other types of parasitic DNA that spreads. And here comes actually one possible explanation why it's good to have a large genome with lots of junk DNA, because that makes that genome less vulnerable to these parasitic forms of DNA. Because if a transposon or a retrovirus would insert somewhere in that genome, if there's a lot of junk, it's very likely that it will just insert itself into the junk and make no harm. So the junk DNA is buffering against parasitic DNA and reduce the damage done by it. And in fact, one has observed in other species like maize, for instance, and the Norway spruce I mentioned, that many types of parasitic DNA and transposons are inserted one into the other in a nested fashion, which demonstrates very nicely that this is indeed a protection against the parasitic DNA. And probably the same thing in animal genomes, where one also has observed this to some extent.
I just can't agree with you on this at all. This I quote Ewan Birney of European Bioinformatics Institute in Cambridge. He states, "Should we be retiring the phrase junk DNA now? Yes, I think the phrase does need to be totally expunged from the lexicon. It was a slightly throwaway phrase to describe very interesting phenomenon that were discovered in the 1970s. I am now convinced that it's just not a very useful way of describing what's going on." This idea of junk DNA was put forth by Orgel and Crick many years ago, before genetics was really on its solid ground as a science. And you criticize the ENCODE project, which has produced a mass of information, new information of genetics, almost the overwhelming majority of which you would be on board with, except this junk DNA idea. When it comes to that, then you're going to criticize them for that. I find that to be specious myself. Why were you be so enthralled to hear about the massive amounts of information coming from this project, but then when they state, well, one of the things we discovered with this is what seems to be junk ain't junk, and then to say, well, that's bad science, but then declare everything else they did to be good science. I find that picking and choosing of the highest order. I don't get it.
Please, Evan, take a look at the literature from those who have criticized the ENCODE project. You will find both those who criticize the conclusions drawn from that project at large, which hasn't been very much and very interesting at all. Some even consider it a waste of money. And you will find intense criticism of this poor usage of the term function, which only serves to mislead people like yourself who is not working with genetics. People draw false conclusions from that inaccurate use of terms.
Well, I would say, here's what I would say to that. I would say, thanks to this ENCODE project, we now know that 84% of the human genome is transcribed. That, to me, is a very useful. I mean, you can be critical all you like of the project because of this one particular inclusion, if you like to, but this junk DNA idea, if you like to. But one thing that it produced was the understanding that 84% of our genome is now known to be transcribed. Now, further genetic study will discover what it's doing, the majority of it, and you may be able to point to a few examples which produce something that seems to have no effect today. I would not be surprised as a Christian and a creationist to discover that the genomes are decaying. And so you'll find that there are pieces of DNA, of genetic information, which are no longer functional or dysfunctional, in some way broken information. But to declare the majority of the DNA to be junk, even to point to this one discovery of the ENCODE project, that 84% of it is transcribed, by itself is a remarkable discovery.
Evan, it was known long ago that a very large proportion of the human genome is transcribed. I don't know the exact numbers, but probably 15 years ago one had evidence for 40-50% of the genome being transcribed at one point or another. So increasing that to 84% just by using more sensitive methods is not a step forward at all.
That's not scientific
It hasn't
advancement.
added any new knowledge about cell functions.
But what it has added is an increased knowledge of how much of our genome is transcribed. That to you is not worthwhile science?
The next step is to see what in this could perhaps be functional. And you already indicated that there are a few small RNA molecules that are important, and they are important for gene regulation, but these are tiny pieces of RNA. They are a minute fraction of what the protein coding genes are. They are highly important, but it's just such a tiny bit of the genome.
Would it not be an advancement of science to discover, say, for example, if we believe 72% of the genome is transcribed by the cell, and then to discover, no, it's more like 84%. That's that's not an advancement in genetics?
Not if you know how sloppy gene transcription is.
What? I'm not sure I understood.
Okay, for the 5th time, gene transcription is not very accurate. There is leakage transcription of genes. Genes are transcribed well beyond the endpoint that is used. No, I don't want to describe this any more time. If you don't want to take in this knowledge, there's no point continuing to the discussion. You can read about this in scientific reviews and in textbooks.
Okay,
Yeah.
so sorry to interfere. Is there anything else?
Yes, I would like to ask him another question.
Yeah, sure, of course. But we have come to the to the state whether we agree to disagree, more or less. So if we could move on, just not only for you guys, or for those 30 people who are viewing this right now. So if we could leave this behind.
Oh, sure,
perhaps,
sure. Leave that subject behind. I'm happy with that.
Yeah, sure.
I wanted to ask him about 2R hypothesis, which he supports according to what he has published, which is the idea that entire genome and block duplications have built the genomes of living things. I would ask, I wanted to, let me give you my view on that, and then ask you to respond to that. I consider the 2R hypothesis to be based on the assumption that similarities between gene families of different species is evidence that they're related over time. That I think it fails to consider responsibly the established understanding that genetic mutation is overwhelmingly degradatory. It fails to responsibly consider that duplicate genes are usually silenced and subject to degradatory mutations, degenerative mutation. It fails to responsibly consider the established understanding that evolution of proteins requires amino acid substitution, which is known to degrade proteins and cause a host of disease. It fails to responsibly consider that a very high number of genes would be lost during block duplication and chromatid recombination in the species that are considered related, and genetic information loss can never be a good thing. It fails to consider the established understanding that duplication and its associated gene losses cause malformation and disease instead of a selective advantage. And it fails to consider the established understanding that the rate of high rate of slightly deleterious mutations in the genomes of living things, including man, create an impossible barrier for the idea that life evolves over vast ages of time. And it fails to responsibly consider that duplication of gene clusters and gene families, which are interdependent with regulatory processes and therefore irreducibly complex, and irreducible complexity discredits evolution. So this idea that this 2R hypothesis idea is an idea that has to basically ignore the fact that all of these things that I just listed are a product or caused by the genetic mutation, duplication of genes, especially blocks of genes, which is known to cause the loss of, at the loci where the genes are spliced, a loss of genetic information by irreparably damaging genes. And so I would wonder, why would you believe that a process known to produce so many destructive effects to the genomes of living things, malformities, diseases, such the list goes on, could be a process which builds the viable genomes and produces healthy creatures that natural selection will then select.
Are you done?
Yes.
Yeah, I have to control myself really now, because I don't think I've ever heard so many misunderstandings and so many misrepresentations of something in such a short time. Have you read well, you probably would need to read a textbook in evolutionary biology to understand the reasoning here. But many of the things you said are complete misunderstandings.
Which one is? If you want to go through them. So what about the idea that block mutation causes a loss of genetic information? Do you disagree with that?
I don't even understand what you're trying to say with that statement.
Well, block mutation is, for example,
what is a block mutation?
A chunk of DNA, entire groups of genes. This is what you put forth as your 2R hypothesis.
No.
But that's what the evolutionist scientists who write of 2R hypothesis argue for. The entire and partial genomic duplications in the genomes of
you said
things.
A moment ago you said block mutation. Now block duplication.
That's what I meant to say. I'm sorry. Block duplication.
Okay, then read the statement again.
Okay, so this process is known to cause a loss of genetic information.
Where does that come from?
I'll give you a source. Yingyuan Liu and Dan Morin state, gene duplication are aberrations of cell division process and are more likely to cause malformation and disease rather than selective advantage. Duplicated genes are usually silenced and subject to degenerative mutations, and regulation of supposedly duplicate gene clusters and gene families is irreducibly complex and demands simultaneous development of fully functional multiple gene and switching networks, contrary to Darwinian gradualism. These two gentlemen are geneticists. And
this was also written in which year?
2000.
Yeah, the 2R concept was not fully accepted then even. And I'm not sure which organisms they are referring to. But the beauty of the 2R concept is that the whole block of gene is duplicated, so one maintains the balance in the biochemical pathways by doubling everything. And the other point is that also preserves the regulatory elements. So you get initially you get two identical things of everything. Then mutations begin to accumulate. Many of the genes are lost. The majority of the genes are lost after genome duplication. But several remain, and they may either subdivide the ancestral genes' functions between them. There are multiple examples of that. Or they may occasionally evolve novel functions. Let me give you one concrete example. Vertebrates started off with two genes for color vision, a blue gene and a red gene, sitting next to one another on the same chromosome. Then the genome doubled and doubled again. So we got 4 copies of one of the genes. That gave rise to the 4 light receptors responding to blue light, UV light, green light, and the dim light that we can see with our rods. So these ancestral genome doublings gave rise to the light receptors that we use in our rods and our cones. And this repertoire exists in birds and fish, confirming that it happened before they diverged from one another. And we know this from sequence comparisons, as well as the chromosomal locations of the genes in the different species. Then in the ancestor of the mammals, the blue gene got lost and the green gene was lost. And later on, the UV light receptor had a few mutations, so it can be used in the blue part of the spectrum. So mammals can see blue with the UV receptor, or the equivalent of the UV light receptor found in birds and fish. The red gene in primates had a local gene duplication that allowed evolution of red-green distinction, but that is a primate feature. These are the only mammals that can have this kind of resolution in the red-green part of the spectrum. And when we look at the gene families involved in vision in rods and cones, it turns out that more or less the whole cascade of components from the light receptors and beyond, and we have looked at approximately 15 different gene families. All of those had additional copies that arose around this time when the genome doublings took place. And we can tell that both from the chromosomal locations in the genome and the sequences of the products.
I would say that your idea that you can tell when this took place is your assumption. But again, I would ask you, this idea of the duplication of genes in small, few individually or in large numbers, as proposed by our hypothesis, is known to produce some very nasty effects in living things. For example, DNA deletion and duplication and the associated genetic orders by Suzanne Clancy, PhD, Nature Education 2008, states that duplications may affect phenotype by altering gene dosage. For example, the amount of protein synthesized is often proportional to the number of gene copies present. So extra genes can lead to excess proteins, because most embryonic development processes are heavily dependent on carefully balanced levels of proteins. Duplications resulting in extra gene copies can therefore lead to developmental defects. Then there's also thalassemia, which is a result of the same process. This is a blood disorder. And then there's a correlation between gene duplication mistakes and cancer. That's well established. I wonder, why do evolutionists believe that processes which are known to produce so many diseases, why would they believe that this is a process that's going to build viable, healthy genomes?
Most of your genes belong to multi-member families. Gene duplication is very common in evolution. And the fact that these massive duplications happened by large chromosome blocks or entire chromosomes, as I said before, was more likely to maintain balance in the biochemical pathways. And there are endless examples of gene duplications giving rise to novel functions. Your whole family of immunoglobulins, for instance, the visual genes I already talked about. One recent example that has been discussed widely is the amylase enzyme genes that allow us to degrade starch, and so on and so forth. Gene family is the rule rather than the exception in evolution.
Evans, are you there?
Oh, okay, sorry. As for the immune system, we know today this is programmed mutation, a set of information, a program in the genome, which creates products on the fly, modifies genetic information in order to combat something that attacks the organism, an irritant, an antigen, to produce antigen. So that's intelligent design, if you ask me, a computer program inside the DNA to on-the-fly modify. So what you're referring to there is, to me, an example of intelligent design. The duplication that you're speaking of is programmed. That's not a random activity. That's not random mutation. That's programmed. That can't be likened to any chance association of molecules, any evolutionary sort of, I don't know why you'd cite something programmed transitional frame mutation and such as evidence of evolution relating to gene duplication.
You are completely wrong. The mutations in the immune system genes happen more or less randomly, and then there is selection among the immune cells in the living individual. That is a selection process.
I don't think the papers would support that it's random, because
I said more or less random.
Yeah, I don't agree, and I don't think the journals would support that. I think the papers in the journals would argue that this is a programmed response, and that's what I've been reading about it. That this is information in the genome designed specifically to modify endlessly something in order to produce to keep the irritant at bay, to keep continuously attempting to block it. That's, to me, intelligent design, not any process of evolution.
The basic setup is a number of copies of segments that can be combined in a certain number of ways. And again, for each one of these, you have several copies to choose from, showing again how common duplications are. Then on top of that, there is somatic mutation going on in these cells as they are becoming mature and selected to be more efficient in the immune system.
That's what I'm referring to.
Both the combinatorial rearrangement of the gene segments and the mutations in the reorganized products. Can I ask you something else? When you say that the Earth is so young, how do you look at variation in the genomes of different individuals, like human beings, for instance? We know now very well how many differences there are between individuals in long stretches of DNA.
Are you talking about human beings?
Yes, for instance.
I'll be happy to address that. But I would first let me mention that somatic hypermutation is programmed mutation process which changes are introduced by nucleotide sequences of immunoglobulin gene DNA during.
Excuse me. I'm not talking about the immune system now. I'm talking about the genome in general, yes, and the differences between individuals in.
Well, I don't see how the human genome provides evidence of evolution. I would say that human beings are a created kind, just like dogs, horses, bats, or any other type of living thing, and we should expect to see variation between the kind. The Creator put enough information in the genome to make it ingenious that it is able to produce variation. The variations that are present between the various tribes of mankind, Polynesian, Celt, Slavic, African, Aborigine, Native American, Asiatic peoples, whatnot, are mere expressions of the genome, the original created genome. There's no anatomical difference between any two human beings on this planet. We all have exactly the same features in exactly the same location of the exact same body plan. If evolution were true, there has to be a change to that anatomy. And that we don't find that in the fossil record. We don't find it in any existing human being. But I would ask you, if you believe that evolution is the various tribes of mankind are a product of evolutionary change, hello?
It seems that Dan has lost the connection.
Yes. Well, just until he comes back, I'll just say for the listeners what it was I was going to ask him. If he must believe as an evolutionist that the various tribes of mankind are a product of the evolutionary process, just like variation within bats or chickens or any other, or fish or any other creature, and that these divergences over time produce a speciation event. And so I was going to ask him what is his opinion on that. Well, there he is. Are you back,
good.
Dan?
Okay, it seems like Dan Larhammar lost the connection, but are you back now? One second.
You may be having audio problems.
Yeah, perhaps. You hear us, Dan? Okay, I will send him the link once again. I think he has it, but I think he already has the link, but I sent it to him once again. So he tried to join, apparently, but there was some glitch there.
I hope he's able to return. This subject of human evolution is very interesting.
Yeah, me too.
I would like very much to ask him about the tribes of mankind.
I know that he uses a laptop. Perhaps if there is some kind of battery problem or something. So I've sent him the link via email. Once again, he should have it anyway, but
right,
just to be sure.
Can't hurt to send it to him again.
Yeah. So well, this is a bit unfortunate. We have 35 viewers, which is good for a hangout.
I was hoping there'd be many more, to be honest with you.
It's me, I don't have that many subscribers. So.
Well, hang in there. As you've said, that I can upload this when it's done as a video on YouTube to my channel too, so it'll get more views there, and that will draw people to your site. They'll find out about you, because I'll put a link to your site under the description.
So will I.
Video. That way, if they want to find your channel, they can click that.
Yeah, so will I. So it's a mutual beneficial there.
Sure.
Okay. Just check once again.
They have such good internet in Sweden, so I hear.
Yeah, we have. So it's quite.
They say Finland is even better. I don't know if that's true or not, but
yeah, they have.
Scandinavia has really great internet speeds, I hear.
Yeah, we have this. We have almost 1 gigabit.
Yeah, better than here in the United States. Yeah. Now you can get that here in the States, but you have to pay pretty penny for it. It's probably more common for people to have that kind of speed in Sweden without having to pay extra money probably to get it.
I mean, if you move into a compartment that has that speed, it's included in the price, so to speak.
Oh, yeah, built into your rent, right?
No. So why isn't he joining? I didn't hear anything that it was anything about him being. What do you say? There was no anger, so to speak, when he left. So
I don't know.
Well, that is what happens when you try to make a. I mean, I know he has my phone number, but I don't have his. So.
Involved in the hangouts that I've done. I've been involved in a lot of them now. I sometimes talk to people from Scandinavia. They usually have better bandwidth than us here in the States. I'm jealous of them usually a little bit. I have to pay more money if I want that kind of bandwidth, so I just have the lowly DSL. It's not very terribly fast, but it's not terribly slow, but it's not real fast.
Okay.
I've had better connections in the past. I had a coax cable connection once, and it was very good. And DSL comes in many flavors. Actually, you can get cheap, crummy DSL, or you can get good DSL. And then there's stuff above DSL. You're probably not even using DSL. What are you using?
No.
Cat 5 cable or Cat 6, something like that.
We are doing fiber optics.
Fiber. Oh yeah. Wow. Yeah, that's the fastest that can be had currently.
So, I mean, I've resent him the link, even though he should have it anyway. And how long have we waited?
Well, I guess it's been 10 minutes, maybe 7 minutes.
5-10 minutes.
No, probably not that many.
No.
5 or 6 minutes, 7 maybe.
That was sad. In a way, I was thinking about starting a post hangout after this one. So yeah. I mean, if
I was going to start one myself. I hope it's not the case that Dan left because he doesn't like the the question I was posing to him about human evolution. It has some very negative historical connotations that go along with it, and maybe he was very displeased with it.
Well, perhaps he was, but I mean he wouldn't just leave. I mean he would say that the discussion was over or something. He wouldn't just leave. I mean,
I don't know. I do know that evolutionists dislike very much the implications of racism when it comes to their theory of evolution, and they really don't too much like to discuss. I mean, see, the idea is this: if human beings are the various tribes of man are a product of the evolutionary process that leads towards speciation, then if you believe in evolution, it's logical to believe that the various tribes of man are different in their divergence from each other genetically, and eventually this process will produce entire new species from the human genome. And one tribe of man may become a different species as another over time. And if that be true, then evolution theory, the natural selection thing kicks in, and you have to figure out which an argument can be made that it's logical. If you believe that, then one species will dominate over another because of their better advantage. And so which of these tribes of mankind do you think hold the key to this divergence that will ultimately produce the one that continues to survive at the expense of the others? There are some very negative connotations that come with this. Perhaps Dan didn't like that and didn't want to respond.
Yeah, perhaps I sent him another email, asked him what happened, and of course I reposted the link. But I mean, if he doesn't join, perhaps we should invite other people to sort of speak about how the hangout went. I mean,
I plan on opening a hangout myself here shortly after this, so I intend to do that myself. You can start one yourself, I guess, if you like, or you could join the one I'm going to start if you like, but I intend to start one on my own channel.
Yeah.
Should we give him a couple more minutes?
Yeah, let's
give him the benefit of the doubt and say his Windows crashed and he's, I don't know.
Yeah.
But it shouldn't take long for a good laptop to boot.
Yeah, say 5 minutes, and then we are at midnight here in Sweden. So
okay,
perhaps.
Okay.
Well, so are you satisfied with this answer so far, or do you have even objections about those, or Evans?
I'm sorry, someone was asking me a question here. I don't leave alone. I'm satisfied that he gave the evolutionist response. Yes.
Mm-hmm.
I disagree with him almost everything he says. The way I see evolutionists doing science is, whenever an assumption can be made, then it comports with evolution theory. But when real scientific discovery produces a problem or causes us to question any part of the evolution theory, then the evolutionists do what they commonly do, and that is throw the evidence and the scientists who discovered it under the bus. And I didn't see anything different about Dan's arguments. He did that at least twice that I could see. Whenever he did it with the junk DNA idea, and he did it with what was the other? I can't recall. Something else, but that's common. It's 3 times I believe he did it. Once was with carbon-14 in dinosaur fossils. Declare contamination. It's always contamination for some reason, or bad science, sloppy science. He actually used the word sloppy once.
Yeah, I heard that one.
Yeah.
Yeah.
I just don't,
Now he has responded. Let's see what he has to say. The connection broken, and he uses sad smiley face. It's kind of funny for a professor. But since I have, excuse me, I'm eating at the same time. Since I've posted
No
the link 3 times to him, I suppose he will join.
Okay.
Yeah, sooner or later, because he had this interruption. I noticed the same thing when he was speaking once or twice. It was kind of breaking up. So
yeah.
Let's hope he.
Yeah, even in Sweden you can have disruptions.
Yeah, it depends on where you live.
It did get choppy a couple of times.
Yeah.
And I got choppy too once, I think, but I don't know if you noticed that. It seemed like it was me, but that was just from my perspective. Maybe you guys didn't notice.
I'm not sure. Maybe perhaps once, but I mean, you don't even use your cam either. So
No, I don't use a cam. It does gobble up bandwidth.
Yeah.
And I just have DSL. I could probably get away with it, but I just really don't have any really interest in putting my mug on the internet too much. I used to, but my arguments come from what I have to say and what I think.
Yeah.
Not what I'm doing or what room I'm in or.
He would be. I mean, if he can email
I'll tell you what,
me.
I'm gonna go get something to drink. I'll be back in about 1 minute. Okay,
Yeah, sure.
Just to see. Okay, won't be but a
Sure.
moment. Okay.
It seems to me that if he can email me, the connection would be fine. So let's give him a couple of more minutes to rejoin. I'm sorry about this, folks, but shit happens. Now I get another email from him. Okay. Okay, so apparently he still has some connection problems, and it's midnight here in Sweden, so even if he would be able to join, it would interfere with his family being sleeping, so to speak. Perhaps I can post this link to some of the peoples in my circles, and they can join. And if NephilimFree would want to leave and start a separate hangout, he's welcome to, of course. So that was a bit unfortunate, but as I said, shit happens. I will talk to Professor Dan Larhammar later on, but I would like to officially thank him for joining this evening or night to speak with NephilimFree, and hopefully he would speak to other peoples too when NephilimFree
Okay,
was
I'm
done.
back.
But yeah, I hear you. So it seems like Dan Larhammar is still having problem with his computer, but he says that it's midnight here in Sweden, so his family has to sleep. So
Okay, all right.
not to disturb them, he would like to call it a night, so to speak.
All right. Well, I sure appreciate you, Evidence for You, hosting this discussion about evolution. Thank you very much. And it's been interesting and very enjoyable for me.
Yeah, thank you.
I appreciate that. I'm going to go start a hangout of my own, I believe,
yeah.
for a while. And this should be uploaded, or you're about to close the end broadcast here in a moment. Is that right?
Well, yeah, I have invited people to.
Well, I think this one should be closed and start another one. How's
that would be better. So it's
that would be better.
This is the discussion between you and the professor is sort of done, right?
Right. Yeah. So why don't you do that, and then and it'll be uploaded as a YouTube video on your channel here, probably in the next 30 minutes or something, and I'll download it. Okay. Thanks again. I'm going to take off.
Yeah. And, thank you for participating.
Thanks again.
Great. Take care. Bye-bye.
Have a good one. Bye.
Bye.